Welcome to Basic Brewing Radio for Thursday, July 30th, 2009. I'm James Spencer. Here at Basic Brewing Radio, we're all about home brewing. Well, this week, Chris Colby, editor of Brew Your Own magazine, joins us to talk about the biology and family history of our favorite critters, brewer's yeast. We know we like what Saccharomyces does, but how and why does it do it? If you're new to home brewing and would like to get into the hobby for the first time, check out our website, basicbrewing.com, where you can find archives of our audio and video podcasts and our DVDs to walk you through basic and more advanced brewing techniques. And you can follow me on Twitter, my username, Basic Brewing, all one word. Also, you can find me on Facebook at facebook.com/basicbrewing.james. And we also have a Basic Brewing Radio and Basic Brewing Video page on Facebook. And if you become a fan of the show there, I'll be sending out occasional notices when shows are posted and such. And also, on the fan page, I had the settings such that if you posted something on the fan page, I didn't see it. So I haven't been ignoring you for the past however long. I didn't see the things, but I fixed that. So I'm going to try to check those and actually interact on that page. I thought nobody was writing in on the fan thing. Turns out I just was looking at the wrong view. I'm old, Thanks again to everybody clicking on the Amazon.com associate link on our basicbrewing.com site. Whenever you think of Amazon, think of us and click on our associate link first. It won't cost you any extra, and you'll be helping us bring you this show. We appreciate your support. There are also associate links for Brew Your Own magazine and the American Homebrewers Association on our site, too. I'm going to be taking a bit of a break from our National Homebrewers Conference coverage this week. I've still got one more awesome interview in the can, one with Sean Paxton, the homebrew chef. I haven't forgot about you, Sean. You're still in the can. You'll be getting out of the can before too long. We had a wonderful chat with Sean. Steve and I did, and Sean's got some inspiring perspectives on brewing beer as food and brewing beer for food and with food, and I'm looking forward to airing that because it's good stuff. But there's a new issue of Brew Your Own coming out, so I wanted to talk to Chris about this week's topic and about our experiment as well. And I'm also hoping to have something special next week to celebrate the goings-on in Utah. This weekend marks the first homebrewing competition in Utah since the hobby became legal this year. And I'm working with Douglas Wierzinski to hopefully capture some sound at the event. So if everything goes well, that's what we'll have on tap next week. So congratulations again to all the Utah homebrewers out there who are brewing without guilt now. I was hoping to be with you guys for this occasion this weekend, but my personal life has interfered, and we need to find another excuse to get together, maybe when it's a little cooler in the fall or something, so that maybe we just get together and brew some legal homebrew in Utah. 4 more states to go. Y'all keep working out there. Let's take a look at the mailbag. Remember that? We hadn't looked in the mailbag in a long time. Larry from Chicago wrote me through Facebook. Larry says, I'm a little backlogged. I know the feeling. But I just listened to the candy syrup episode. Larry says, using a similar process minus the ending water and using a pinch of acid blend rather than that other stuff, I make hard Belgian candy sugar in a fondue pot that has the electronic temperature control. This way, you don't have to watch it. Just let it sit about 270 degrees for a couple of hours or until you get the desired color. Then bring it up to 300 and pour into a cookie sheet lined with parchment paper and let it cool. Break it apart and store it in the freezer until you're ready to use it. Larry says he liked the idea of using a fondue pot that was collecting dust, and it's non-stick, which is perfect. That's awesome. I love using old appliances for new uses, like my old hot air popcorn popper that I use for roasting coffee. I love that. You get these what are supposedly single-use appliances for Christmas or gift giving or whatever, and then they sit taking up space in the cabinet. So it's awesome that we can find new uses for those old things. Travis from somewhere in the U.S. writes, I know you ask people every week to click on the Amazon link, but I used to forget until I put a copy of the Stepping into All Grain video in my Amazon shopping cart and clicked Save for Later. Now whenever I check out, I see the video there, and if I forgot to click on the Amazon link on Basic Brewing first, I close out of Amazon and log in through the link on your page to give Basic Brewing a little support. Works for me. Well, thanks, Travis. I didn't know if you might make this suggestion about putting the video reminder in the cart to your listeners. It's the least we can do for all the info and entertainment you provide. Well, that's nice, Travis. I also enjoy hearing you read my obscure purchases out on the air. I was the guy who bought such things as the 5 love languages, backyard chickens, and baconaise. And that's not a P-34 or whatever flying outside. If you hear, that's a lawnmower. Live from suburbia. Baconaise. Wow, thanks, Travis. It's a little self-serving to read that letter, I realize, but I think it's funny that I've read so many things, so many of his things on the show. A bunch of stuff goes through that link. Lots and lots of things go through that link, and I have no idea who buys what, as I say at the end of the show, so it's funny that I picked several of Travis's things at random. You have great taste, Travis, and I appreciate your support. Myron in Iowa has a good constructive point of criticism. Myron says, you have a very good show, and I listen regularly. Thanks, Myron. That's not the constructive part. Myron says, I would request that you preface your interviews with an explanation about what the process exactly is. I'm 20 minutes into the show, and I don't know what a decoction is. It is basic brewing radio. I have noticed this before in your show. Well, thanks, Myron. That's a very good point. We did the decoction show last week and kind of assumed that everyone knew what decoction was, and that's my bad. I'm sorry about that. For those of you still confused, decoction is the process of taking a portion of the mash, boiling it, and returning it to the mash tun to increase the mash temperature to the next mash rest if you're doing a multiple temperature mash. It's an old way of doing things, but as you can tell from the past episode, many brewers still swear by it. And it's fun, too. And I think there's a DVD somewhere that shows you how to do it. Scott from Manassas, Virginia writes, after listening to our December 11th, 2008 episode on smoked beers, Scott says, one thing not mentioned as a way of smoking beer is to age the beer in charred wooden barrels, or for the home brewer, wood chips. I'm planning a pale ale aged on roasted apple wood chips. I'm aiming for a crisp apple but smoky flavor. Wow, that sounds tasty. Appreciate that idea. James, formerly of Tennessee, who's written to the show in the past, and we've read those emails. James says he's got a gig at Schlafly in St. Louis giving brewery tours. And so he's all excited about that. I am too. We can get Schlafly beers if we drive an hour north into Missouri. And they have some tasty brews. Congratulations, James. If you take a tour up at Schlafly and you see a tour guide named James, give him the Basic Brewing secret handshake and tell him we said hi. It's not really a Basic Brewing secret handshake. We got a lot more good stuff in the mailbag, and I appreciate all the emails. And we should have time next week for more. And I apologize if I've been slow in getting back to you lately. If you've sent me a note, being out of town and blah, 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 insert your excuse here. But I appreciate your patience. I'm all caught up now. I'll be behind again tomorrow. But anyway, let's get on to our interview with Chris Colby. Put on your science hats, everybody. What makes brewer's yeast so helpful? And how did it get that way? Well, Chris Colby, welcome back to Basic Brewing Radio.
Hey, James. Thanks for having me on the show.
It's been a while, but both with our little vacation in June and then all the episodes that we've been having from the Homebrewers Conference out at Oakland. We've had a little break, but it's good to have you back. I want to start off with a letter that is pertinent to our discussions today, or at least part of them. This comes from Andy, and Andy didn't say where he's from, unfortunately. He said he's a new listener. He said he didn't participate in our experiment of leaving the beer on the primary yeast. He says he didn't participate because he hadn't found the podcast at the time he started listening. But Andy says, by accident, I conducted the experiment. I brewed an IPA on April 9th, 2009, and because of a very busy schedule, I didn't rack it from the primary fermenter until July 2nd, 2009. The beer sat on its yeast cake for 84 days. And Andy says it had no ill effects that he could identify. Andy says, we drank most of it on July 4th weekend, and everyone gave it good marks. Needless to say, this will not become my new process, but I just wanted to contact you with this information. So there you go. An IPA sat on the primary yeast for 84 days, and it didn't screw it up, apparently. So there's more data to support our findings of our experiment.
Yeah, I do a lot of accidental experiments that way. Yeah, I had an IPA recently, too, that I left quite a while on the yeast, but unfortunately I didn't have a control group for it, because I actually thought that one did, because that one was left way longer than in our experiment. I left that one for a couple months. Mm-hmm. The results of our experiment were interesting. I thought they were, worthwhile.
Yeah, the one thing that's missing in his experiment was, like you said, there's no control. There wasn't another fermenter that he bottled on time or packaged on time. So we don't know if he had racked the beer off of the primary yeast at the regular time, if it would have made a difference in the flavor of his beer. It's just that after 84 days, the beer wasn't ruined. So
right. Yeah, you don't know how much better the beer would have been had you racked it.
Right. But it's interesting. I mean, and if anybody out there is listening that has data to the contrary, please let us know, because we're not just interested in those that are preaching to the choir here. We want to hear evidence on both sides of the issue. And speaking of issue, the story on the experiment, the yeast experiment, is going to come out in the upcoming issue of Brew Your Own magazine. And I thought that turned out real well. It's a collaborative effort between us two, and we list the data that we collected from the 12 home brewers around the world that participated in the experiment, and quoted some of you guys. So you may see your name in a quote in the article. I thought it turned out well.
Yeah, I liked the article. Our technical editor, Ashton Lewis, who's also Mr. Wizard, also liked the article. Although he, in that very column, his column this month, he actually claims that you should do the exact opposite.
So it'll be interesting to see if people start kind of modifying their or footnoting their advice from now on. Rack the yeast or beer away from the yeast as soon as possible, or maybe not,
or don't. Yeah. I think his point was he's as a professional brewer, he's trained and sensitive to those flavors, and he can detect them and doesn't like them. Mm-hmm. And where our experiment showed more or less that you didn't, with a couple weeks extra yeast contact, you didn't absolutely ruin the beer, although we did taste a difference. Mm-hmm. And then from there on, it's if the difference is something that agrees with you or not, or that you think is so not completely incompatible. I mean, I think the basic idea of ours was to address homebrewers who, like the letter writer said, due to time constraints, he forgot to rack it. Happens all the time in homebrewing. And on the internet boards, you read all the time, like, oh, I forgot to rack mine on time. Should I dump it down the drain? Mm-hmm. And our results sort of strongly say, no, it's going to be, only slightly different than what it would have been had you racked it on time.
Right. And if they are experiencing some severe off flavors at that point, it could be that there's something else going on in the brew rather than just leaving it on the yeast.
Right. I was amazed at how consistent the experiment was across all the different people who did it. We tasted it, you, me, and Steve, via Skype. The differences there were tiny. Mm-hmm. The rest of the people who did the experiment, that seemed to be, there's a few people said there was no difference. There's a few people who said there was a slight difference. And, a few others said that there was a more pronounced difference, but none of them said, the racked promptly beer is good and the, racked later beer is wrecked. Mm-hmm. So it was good to see, across a variety of styles, across a variety of ale strains, that the consistent results came out of that.
And we've decided on the next experiment in the series of experiments, collaborative experiments between Brew Your Own magazine and Basic Brewing Radio, and that is...
How does your pitching rate affect your beer fermentation?
That's right. It was one of those that was suggested by a Twitter user, and I could find that if I could find the piece of paper that I wrote all that down on, but I'm sorry I don't have that with me. But it's a question that I've thought about for a while. And specifically what I was thinking is, can you overpitch?
Sure.
But there's another side of that, and can you underpitch?
Yeah, and we've got we haven't completely settled on it yet, but we've got a nice idea for an actually very nice, tight experimental design, and one that'll let us collect both some very objective data, like how long till your fermentation starts, how long does your fermentation go before it's finished, what's your final gravity, and also combine that with more subjective, first some objective tasting data, just can you taste a difference? Mm-hmm. That's more objective than subjective, and then followed up with, do you like one or the other? So yeah, I think this is going to be a good experiment, and hopefully we'll get people to participate in it, because it should be something that, equipment-wise, all you really need to do is brew a single batch of beer and spread it out between 3 carboys, mm-hmm. and then pitch each at a different rate. So we'll hope to get a lot of participation and get a nice clean result that we can give a result with confidence to homebrewers.
So stay tuned to Basic Brewing Radio, and also check Chris's blog on byo.com, and we'll be announcing the details of that when that is going to happen, when we can work out the process and make it easy enough to follow, and hopefully fairly foolproof in being able to collect some reliable results. So, yeah, I'm excited about that.
Yeah, me too. This will be a good one.
Well, the meat of our discussion this week is an article that you wrote, in another article that you wrote in this month's issue, and that is on the biology or the evolution of yeast. And this kind of plugs into your personal background, right? Your educational background.
Yeah, I was a biology and chemistry major as an undergrad, and then my graduate work was at Boston University in evolutionary biology.
So this fits right in.
Yeah, I was actually sort of excited to do this story. I hadn't really looked much at any of the literature of yeast evolution in a long time, and there was a lot that I'd missed in the last 10 years.
And what we're talking about is basically 2 or 3 specific strains, just a small hunk of the wide range of yeasts that are out there, right?
Yeah. One of the interesting things, I think, when you think about brewing is there's basically 1,500 species of yeast, give or take in the world, and there's tens of millions of species of bacteria. And yet, amongst all those, potential microbes that could ferment, all we really have is Saccharomyces cerevisiae, which is ale yeast. We have Saccharomyces pastorianus, which is lager yeast, and that used to go by a bunch of other different names like Carlsbergensis and other stuff. But if you throw wine into the consideration or into the conversation, you could include Saccharomyces bayanus. And a very few breweries brew with Brettanomyces, but that's a finicky yeast to brew with. You got to pitch a ton of the yeast, and it's always a prolonged fermentation. So it's interesting to think of all the possible microbes in the world. How come there's only basically two species that are worth, or that are good brewers.
And yeast is a fungus, right? It's kind of like a single-cell mushroom or something like that.
Right. Yeah. Yeast belongs to the same group as fungi, which are mushrooms, molds, yeasts, and other related things like that. It belongs to a group called the Ascomycetes, which is the sort of common name is the sac fungi, SAC fungi, and there's about 30,000 species of Ascomycetes, and of them about 1,500 are yeast. And of them, there's, a handful of different Saccharomyces species, Cerevisiae, Pastorianus, Bayanus, and then just there's a bunch of other ones that aren't really used in brewing, but they're still, related.
So what in the wilds of the world, when they're not working on a batch of wort, yeast, brewer's yeast, and yeast in general, what do they do?
Yeah, yeast in general, if you talk about just any generic sort of yeast out in the environment, they, for the most part, they live on simple sugars. If you're talking about Saccharomyces specifically, although not necessarily just the brewing strains of it, but the wild strains, they live on fruit. If you've got a fruit orchard or whatever, if you were to go anywhere where there's a little lesion in the fruit, like a little cut or something where the fungi can get into the fruit and start fermenting the sugar, that's where they are. So they live on, 9 times out of 10, a Saccharomyces yeast is going to be, somewhere on the outside part of a fruit eating its sugars.
So if you pitched, say, some fruit that had some activity on it into a wort, you might get some fermentation, but it might not be the kind of fermentation that you want, because you may be getting some good brewer's yeast, and you may get some of these other wild yeast as well.
Well, one interesting thing, if you crushed up a bunch of fruit, like, for example, wine grapes, and just let them go on their own, you would start off with a fermentation of a lot of different bacterial species. You'd have some Enterobacteria starting out. You'd have Saccharomyces would be in the mix, and you'd have lactic acid bacteria and all that. What you'd find was that rapidly the Saccharomyces yeast would take over the fermentation pretty quickly, and they would kill off most everything very quickly, and then they'd be one of the few to run to the end. I mean, there are some wineries who do. They usually, it's not exactly this way, but they do sort of use natural fermentation. So yeah, if you were to pitch, just take some fermenting fruit and add it to a beer, you would have a shot at getting some sort of fermentation. I mean, I did it recently with some friends of mine from the Zealots. Joe and Jim and I made a recreation of a beer brewed in ancient Samaria, a beer described in the hymn from Ninkasi. And what I did is I crushed up some dates and some grapes, and allowed that to start fermenting on its own. I also spiked it with some dregs from a Rodenbach Red clone too. But it was started with a mixed fermentation, and we fermented a beer with that. And so you can do it, but your results are going to be they're not going to be sort of reproducible. If you tried it 10 times in a row, you'd probably get 10 different fermentations, with some being quite nice. Although if you start with a wild fermentation, you're going to have some lactic bacteria in there and some sourness, but in some cases you might get a speedy fermentation with a nice light sourness, and sometimes you might get an incredibly sluggish fermentation and just have this horrible phenolic flavor in it. But yeah, you can, wild Saccharomyces species, cultured from rotting fruit, can lead to fermentation.
So you said that during this fermentation process, the Saccharomyces yeasts outcompete their competitors. How do they do that?
They do that via alcohol. They just poison them.
Winning through alcohol.
Yeah, just flat out poison the other. Most other species cannot other species of microbes, whether they be bacteria or yeast, just can't deal with ethanol. And Saccharomyces is a strong fermenter. It gets in there and really starts turning sugar into alcohol in a big hurry. And this changes the playing field in a fermentation. Say outside, in a cut in a tomato or something, from a bunch of species initially, whatever's landed on it in the air, to very few in a rapid hurry. Like Lactobacillus and Pediococcus and a few other strains can tolerate a little bit of ethanol, but most yeast and bacteria will just die.
So what's the difference between the strains that are good for brewers and just the wild yeasts in how they approach that fermentation process?
Well, there's a lot of different differences, and one of the most interesting ones, though, is that if you look at how yeast will utilize sugar, a non-Saccharomyces kind of yeast that, say, lands on a piece of fruit or whatever, it's going to land there. There's going to be the simple sugars from the fruit, but there's also going to be oxygen because they're just on the outside of the plant or on the outside of the fruit. And what they'll do is they'll break down the sugars by aerobic respiration. Okay, they'll take it in, they'll convert the glucose through, they'll feed it through the glycolysis pathway, which, if you remember your high school biology, that's how glucose gets broken down into 2-pyruvate molecules through a series of steps. Then from pyruvate, it'll get fed into the Krebs cycle and degraded all the way into carbon dioxide and water, and releasing quite a bit of energy. An interesting thing about brewer's yeast, Saccharomyces cerevisiae, even the wild strains of it, is when they hit fruit, rotting fruit, they've got the same sugars available to them. They've got the apparatus to break it down aerobically and yield all that energy, but they break it down via fermentation, which yields far less energy. But as we've seen, it also produces ethanol. So they originally start getting less, bang for their buck out of their food, but in doing so, that they, poison the environment around them so that they can get all the food in the end. And they also do one neat thing is in the wild, when they're fermenting and there's still oxygen available, once the ethanol level rises to a certain point, to a point that pretty much everything else has died off, the yeast actually start taking the ethanol back in and they'll consume it. They'll change ethanol back into acetaldehyde, and then through a series of steps, they'll turn it into a derivative of acetate, acetyl coenzyme A, and then they'll feed that into the Krebs cycle and degrade it completely. So they initially switch to fermentation to poison their competitors, and then later on, if they're fermenting and there's still oxygen available, they'll start taking in some of the ethanol that they've already made and consuming that and getting their energy from it.
We hear about acetaldehyde as a bad thing in the brewing process. What kind of off flavors does that produce?
They say acetaldehyde gives the little green apple flavor, and that's pretty accurate. It's, if you smell it, it's got a distinct smell. It's actually the smell in a person's breath when they're drunk is not ethanol, which it's hard to smell, and it's got a very lightly sweet flavor. It's acetaldehyde is what you smell on a drunk's breath.
So the way that brewer's yeast ferments in the presence of oxygen, producing this off flavor component, is yet another reason to keep oxygen out of your fermentation after it starts, right?
Yeah, acetaldehyde is an intermediate in yeast metabolism in fermentation. It's pyruvate is converted into acetaldehyde, which is then converted into ethanol. And in a brewery fermentation, there is acetaldehyde that gets made in the yeast and escapes the cell and becomes part of the flavor profile of beer, although most brewers strive to minimize that. But yeah, if you were to open your your fermentation to oxygen at that point, amongst the sort of host of problems you would get, including staling and potential for diacetyl to form and stuff, would be the potential for some more acetaldehyde to form. But normally in a brewery fermentation it wouldn't, because at the time the yeast would, the time that a wild yeast strain would start taking in ethanol and start consuming it, in a brewery fermentation the oxygen's been excluded, so it can't take the oxygen back, or the ethanol back in and feed it through the Krebs cycle, because the Krebs cycle requires oxygen to work.
So part of your research for the story was looking into papers that talked about the evolution of Saccharomyces. How did Saccharomyces get to be such a good friend to the brewers? What's the history behind that?
Well, there's basically 3 different big steps, and then there's an extra one if you're talking about lager yeast. The first big step for Saccharomyces was duplicating its entire genome. A genome is just a word for like every gene that you have on your chromosomes. And so the first step, there was a complete genome duplication, or a whole genome duplication as they call it. There was a second step where they simply a small part of their genome got duplicated, and it happened to be the part that had alcohol dehydrogenase. And the third part, which is just an incredibly simple step, but means a lot to us, is there was the deactivation of a certain gene that led to off flavors in beer. And then if you want to talk about lager yeast, there was the additional step that lager yeast are actually a hybrid species. They've got the genomes of more than one kind of yeast in them, which explains at least partially why they're good at doing what they do.
So what does this mean to us? I mean, how do these duplications and modifications in the chromosomes, how does that result in the products that we get today?
Well, the interesting thing about it is, I mean, we've, as brewers, by trial and error, initially, and by, scientific research later, we sort of learned how to handle brewer's yeast. So, I mean, by learning that, as scientists fill in the evolutionary history, is not really helping us directly be better brewers. It's just giving us a nice appreciation of what has gone before. But there's some interesting, for the brewer, there's interesting aspects to all of these different steps. Like the step in the whole genome duplication, what happened is the ancestor to Saccharomyces cerevisiae jumped from having 8 chromosomes to 16. But then it had a duplicate of every single one of its genes. And what scientists found out was that very rapidly those genes began being deleted or inactivated or whatever, till pretty much the modern species has basically the same number of genes again, with only about 10% excess. So the whole duplication, at one point it had 100% excess genes, a different copy of every single gene. Now it only had about 10%. And what they found among those 10%, most of those were genes that dealt with sugar utilization. So, and it's interesting when you look at those genes where the 2 copies were retained. One of the copies stayed pretty much the same. If you look at closely related species, there's a analog to that, or actually a homolog to that. It has very similar sequence, very similar activity. But in Saccharomyces, the other one that they retained frequently has diverged in sequence and function. And so we know that through basically looking at how many different changes there have been and knowing mutation rates and stuff, they calculated a rough time of divergence from Saccharomyces and other species of yeast, closely related species of yeast. And they know the whole genome duplication occurred pretty much right after that. And they found out that was about 100 million years ago. And the interesting thing about this is that they found out Saccharomyces doubled its genome size and retained all those extra sugar metabolism genes right about the time that the angiosperms were arising. Angiosperms are flowering plants that make fruits and have all the sugar, and that's where Saccharomyces lives on today. So that's kind of neat. You have some hint of where because certainly being able to handle sugars is something that brewery yeast are expected to do. The second step I talked about, the ADH duplication, is interesting to brewers because now you have two copies of alcohol dehydrogenase. One that they call, cleverly enough, ADH1, converts acetaldehyde to alcohol, the final step in fermentation. But the other one preferentially converts ethanol into acetaldehyde, and that was a necessary component of yeast being able to both excrete ethanol, as a poison and later consume it and get back some of that energetic value that they've lost by not, aerobically respiring it. So that's of interest to brewers. And, the final step with the POF is, or the gene that deals with off flavors is called POF, or phenolic off flavor. And it, in an intact copy of the gene, of the kind that you'd find in most wild species, it produces 4-vinyl guaiacol, which is the sort of spicy note that you have in a German Hefeweizen. Mm-hmm. But in other beers, you don't want that. And anyway, in brewing strains, that's just deleted.
Now, the Mother Nature, I guess, had a hand in helping along with the differentiation back 100 million years ago with the appearance of the fruit-bearing plants. I guess that you could say that the lager brewers in storing their beers cold might have had a hand in establishing the prevalence of the lager strain of the yeast, right? I mean, this is an instance where human interaction sort of was, instead of natural selection, we had human selection in sort of differentiating this strain as well.
Sure. When you look at lager strains, and people have always assumed the lager strains arose in breweries. Even before anyone had any evidence of it, it just sort of made sense. And anyway, what scientists have done recently is they've taken a look at the genome of lager yeasts, and there's actually two different kinds of lager yeast out there that, in one of the papers at least, they cleverly call Group 1 and Group 2. Yeah, those scientists.
That's why they got the degrees and stuff.
Yeah. In Gregory Noonan's book, he calls them the Froberg and the Saaz type lager yeasts. And actually, I'm trying to talk to some people and try to get a map of what these people are talking about mapped onto what are the Y yeast numbers and the White Labs numbers are. But anyway, when they look at the genome of modern lager yeast, what they find is there's an almost complete genome of the species Saccharomyces bayanus in there. Saccharomyces bayanus is a wine. It's used with certain kinds of wines, and it's a good low temperature fermenter. And so that's one of the components in lager yeast. And the second component is sort of the remnants of Saccharomyces cerevisiae genome. There's whereas the Saccharomyces bayanus genome is almost complete. There's just sort of random remnants of cerevisiae. And in the two different groups, there's one that actually has sort of two copies of a Saccharomyces genome in it and one bayanus, and there's one with one Saccharomyces and one bayanus. And one thing that researchers did is they said, okay, where did these Saccharomyces cerevisiae sequences come from in lager yeast? And so they took the sequences from lager yeast, they sequenced several strains of ale yeast, and then they sequenced sake yeast, wine yeast, and these are all cerevisiae species, and they sequenced some cerevisiae sampled from like tree sap and fruit fermentations in the wild and stuff like that. And they found out that the cerevisiae derived sequences in lager yeast come from ale strains. And in fact, they found out there's two different ale strains, fairly closely related, but not clearly different. So it's pretty clear that the species Saccharomyces pastorianus arose via hybridization in a brewery fermentation.
Huh.
Something that, back in the old days, people didn't ferment with single strains. They were all mixed, and there was probably, wine strains mixed in with the beer strains, and, you just whatever mix of stuff you had worked. Certainly in Germany, where lager brewing started, they also have a wine tradition there, and they ferment white wines at cold temperatures. So, those strains were about. And anyway, the evidence very, very strongly suggests that twice in the history of brewing, and this only goes back a few tens of thousands of years at most, and then for lager rebrewing, you're talking about much, much sooner, or much, much closer to current time, that, sometime in there, two different times, a hybrid between, Saccharomyces cerevisiae and Saccharomyces bianus occurred, with a subsequent, as the species kept evolving to become a better lager brewer, it kept losing little bits of the cerevisiae genome.
So it makes me wonder if that happened in the lagering vessels of old. It makes me wonder, with all the experimentation with all the critters nowadays, with especially American craft brewers, I wonder what's on the horizon. What kind of weird bugs are going to come out of that?
You can select for all sorts of weird stuff in laboratory studies they do with all kinds of different groups of bacteria, different groups of yeasts are more or less susceptible to hybridization. They're more or less susceptible to, in bacterial species there's little pieces of DNA called plasmids that are often shared across species borders. There's ways that bacteria can put a little bit of its DNA into another bacteria. There's, certainly in yeast, which have a sexual part of their life cycle, a lot of times in non-brewing strains, they can mate with other closely related species. And yeah, if you were to look and, if you sampled, sort of brewery yeast from every brewery fermentation that ever happened and, went in and sequenced it, I'm sure you'd find some interesting stuff.
Well, this is interesting stuff. I love all this science stuff. And if those out there are interested in reading more, they can look at your blog, right, and find out a list of resources they can go to.
Yeah, the story in BYO is really short, and there's a lot of information out there, and it's the kind of thing that's up my alley and I'm interested in. So I'm going to, on my blog, just have a series of, quick entries where I'll give you, list a couple scientific papers that found, a specific result, just a brief discussion of, here's what they found, here's how they did it. And, just to give interested people, jumping off point into the scientific literature to check this out, because if you're interested in biology and brewing, I know you're probably going to think this is pretty cool.
Well, awesome. I appreciate it. It's a good article, and it's cool to jump off into the kind of deep end of the scientific information every now and then. It's not necessarily practical information around the brew pot, but you never know when this kind of stuff might come in handy later on down the line.
Yeah, it's there's not a real big practical implications of this, because brewers have been using the yeast for thousands of years, and they've by trial and error and later on by a little bit more scientific approach have figured out how to use it. But the learning the background info, it might lead to some interesting stuff. Like, I know there's a group of scientists in Japan who they made a yeast strain, a brewing yeast strain, where they knocked out the ADH2 gene, the gene that converts ethanol back into acetaldehyde, and they found out that genetically modified yeast produced beer with less acetaldehyde in it. So, certainly finding out what genes a given species has and what its metabolic pathways are, are going to help when people want to go in and genetically modify yeast strains for better fermentations, or at least fermentations more tailored to what they're shooting for.
There you go.
There you have it.
Better living through science.
Mm-hmm.
All right, Chris. Appreciate your time.
Thanks for having me.
Well, thanks again to Chris. You can get a free copy of Brew Your Own magazine by clicking on the banner ad on basicbrewing.com. And if you decide to subscribe after reading that issue, you'll be helping to support this show. And thanks to everybody who's done so. A bunch of people have. Appreciate that. And by the way, Andy, whose letter I read at the start of the interview, is from San Marcos, Texas, which he says is just down the road from Chris. So that's cool. Remember, we've got brewers' logbooks and are just literally a handful. And our new basic winemaking, well, it's not really new anymore. I didn't need to change my little script. And our basic winemaking introduction to wine kits DVD out there in our online store. If you have brewing questions, show suggestions, or just want to say howdy, write to james@basicbrewing.com or just fill out the contact form on basicbrewing.com. And don't forget to tell us where you're from. You can also talk to me on Twitter and Facebook. You can check out our low-tech lagering and decoction mashing DVD on basicbrewingshop.com, along with our other DVDs as well, Introduction to Extract Home Brewing and Stepping into All Grain. And we've got combo deals to save you a few bucks on our store. And you can see a listing of the fine folks across the country who sell our DVDs on basicbrewing.com. And if there isn't a homebrew shop, a vendor in your area who sells them, you can order them directly from us online in our online shop at basicbrewingshop.com. We've got shirts of different colors, and thanks to everybody who's continued to click on our Amazon.com link. We should appreciate the support there. Our featured products this week that were purchased through the link are chemical-free hard red wheat seed, 5 pounds, plant and grow wheatgrass, flour, grain, and bread. Emergency preparation food storage. Excellent germination. And round cracker keeper. I just like the sound of round cracker keeper. Thanks, everybody. Remember, can't tell who bought what, so no worries. Just I wonder if Travis bought the round cracker keeper. Just click on the Amazon.com logo on our site the next time you feel like Amazon shopping. And we appreciate your support. And don't forget, you can also join the American Homebrewers Association through an associate link on basicbrewing.com. That's all until next week. Until then, thanks for listening. I'm James Spencer. Production help for Basic Brewing Radio and our website is provided by our buddy Kelly Dotson down in Austin. Basic Brewing Radio is a production of Active Voicing. We'll talk to you next time, everybody. So long.